Merced. No problem. So the title of my talk today will be about wildfire effects on watersheds across the Western U.S. Using remote sensing. So there's an estimated over 1 million people in California that lack access to clean drinking water. And according to data from the California State Water Resources Board, a majority of them reside in the Central Valley. Let me change the, so it's better for you. Sorry about that for those online. Slide right here. Okay. No?
You might, yeah, you should.
Okay. I'm gonna try to do it right here. Hopefully that, I'm sorry if that's gonna get in the way of it. So that's primarily located in the Central Valley, and the Central Valley is historically underrepresented, and also the lower income bracket in California. So while I acknowledge that a lot of this contaminated water is typically talking about groundwater, and the terms of like the water that I'm talking about for my project, I'm looking at surface water. So even the majority of the groundwater used for the state is what's contaminated, it just basically means that the limited water available just makes it that much more important. So the limited surface water is going to be much more important in years to come. So about 30% of California's water supply is stored in the Sierra Nevada snowpacks, and that can be shown in the top left. So on the top left part of that picture is in late March, so you can see that the snowpack is completely full. And while in early July, it's already dwindled down to almost nothing. So with climate change, we're starting to see that snow melt is actually occurring estimated 1 month earlier than usual. And according to the California Climate Assessment, it's determined that about 1/3 of the large reservoirs, so the two largest reservoirs in California is Lake Shasta, shown in the top right, and in the bottom right for Lake Oroville. It's predicted that about 1/3 of annual storage will be kept in these reservoirs by the end of the century. So while many are concerned over the actual amount of water available, what's often overlooked is the actual quantity, the quality of water. So it doesn't really matter how much water we actually have if it's not really usable, right? So even though we have water treatment systems, this might not exactly be feasible in the lower income areas as mentioned earlier. And especially with more water treatment systems, it might not be feasible if reached to a certain water quality limit. So with higher temperatures and more extreme unpredictable weather patterns, we're going to be able to predict a more variable water supply and water quality conditions across the world. So according to the IPCC Annual Report 5, they predict that there's going to be a reduced water quality, which even with conditional water treatment, it might not be able to meet those certain water quality thresholds. So when we're talking about the water cycle variability, we're talking about more increased precipitation, whether it's snow melt or more frequent and longer droughts. It's also different river flows, and all in the end, which would affect water quality. And according to the National Climate Assessment in 2017, they've predicted that water quality is primarily going to be changed due to these mobilizations of sediments and pollutants that, again, that is affected due to the different water cycle variabilities. So also, as previously mentioned with drought, it's expected to become more frequent and longer lasting. And these types of the coupling of these, so temperature, soil moisture, humidity, and fuel availability will lead to increased fire risks, right? So the lack of rain and coupled with low humidity, it will dry out the trees and vegetation, providing fuel. And on the left, provided by NASA's Earth Observatory, it shows how extreme and exceptional drought is actually increasing over time. as well as on the right, it shows that wildfire, the extent is increasing over time as well.
Brittany,
Yes.
If you hover over those pictures from the online, there should be
Oh.
a collapse button,
Let me see.
and then we can actually see the whole slide. So just hover, see how up in the top left, there's grid.
This?
Yeah, click that one.
Okay.
There you
Oh,
go.
perfect. Okay, great. Thank you. Let me just also put this in the top right here. Cool. Thank you. So yeah, you can see that the wildfire extents are increasing over the years. So here are some bar graphs from data collecting from MTBS. And this is showing that even though in the past couple of years or past couple of decades, we don't necessarily see an uptick in the number of fires in California, but in terms of the actual area burned, it is increasing. And another thing just to note that MTBS often frequently updates their data. So this data, I mean, I just did this last night, but in a couple of months or even next year, these overall quantity can change. But as this supports the previous image, even though we're not seeing more fires per se, we're seeing more area burned over time. So again, I'm talking about California. So in the PPIC, which is the Public Policy Institute of California, they asked California residents, what do they think are the most important environmental variables or issues currently happening? And everyone or most people basically mentioned water supply or wildfires, right? When you think of California and you think about environmental issues, these are two topics that most people tend to think about. And when they were individually asked about water supply, a good majority of them did agree that water supply is an impending risk for the state. And with that, we've seen a lot more news coverage talking about wildfire and the relationship it may have with water quality. So on the right images, I have two pretty recent fires in California. And in green, I've highlighted where the closest reservoir is. So you can see that these are two pretty big fires, and it actually has a decent amount of high severity burn. And you can see how close in proximity they are to local water supply. So basically my overall project is trying to determine what the relationship between both wildfire and water quality is. And as previously mentioned, I'm tending to use remote sensing for my work. And the reason is because the current research is super limited geographically, temporally, and it's typically only done in a few or clustered lakes. So just off beat for geographically, most of the papers that I've read talking about this topic typically is on the Midwest. So looking at some in Colorado, a lot in Montana, or they're done in boreal Canada. And for temporally, you're only able to actually do these field campaigns for a limited amount of time, right? Like feasibly due to budget restraints or just to the length of the project, they're only able to look at these lakes for a very limited amount of time. While with remote sensing, you're able to do that since the beginning of the launch of the satellite and until this ends. So for decades. And single or clustered lakes. So on the top right, I'm showing the Great Lakes, which are often done a lot of water quality monitoring. So especially Lake Erie in terms of chlorophyll detections. But in my case, I'm looking at California as a whole. And so far known to me, there is no actual work that has been done on a statewide level for this specific approach, asking this type of question. So an overall satellite and variable overview. So the three variables that I'm using to indicate for water quality, so they can only be determined through remote sensing, right? So they're all optical properties. So first is chlorophyll-a, which is found in all like plants for photosynthesis. And the other is cyanobacteria. So cyanobacteria has chlorophyll-a. So when you're detecting chlorophyll-a, it might be cyanobacteria. However, cyanobacteria is unique. That has a certain pigment called phycocyanin that could only be measured in a very specific wavelength of about like 680 to 700. So the only satellites that are able to measure it are Envisat's Merced and also Sentinel-3's Ocean Land Color Imager. While for Sentinel-2, you're able to monitor chlorophyll. And for Landsat, you're able to monitor turbidity, which basically is the measurement of how clear the water is. And that's important due to certain sediment that's suspended within the water can also carry some pathogens, which may induce illness. And also, turbidity is very expensive to clear out through normal treatment practices, whether through coagulation or through actual filtering. And here in this table, I'm providing an overview of each of the satellites, their visit time, the spatial resolution, and which variables they measure, and what years you can measure for each.
Brittany?
Yes.
Can you just provide some clarity for some of us who aren't water people?
Of course.
Why is cyanobacteria bad?
So cyanobacteria, like trying to look at that aside from chlorophyll-a. So chlorophyll-a, like that's how we typically think of like algal bloom. So like, you know, that green scum. So cyanobacteria is specifically more important because it can contain cyanotoxins. So there's like 3 types. One is microcystins. Another is, I can never pronounce it. It's like Anapha something. Like they're very long, you know, biological names. But these can induce illness if ingested. So there have been reports of like animals dying if ingestion, or children being in hospitals if they've been drinking this water. So I'm not saying exactly like, oh, all cases, if you see cyanobacteria, you will be ill, but with greater concentrations of it, it could become an actual public health hazard. So that's why cyanobacteria is particularly important. And again, chlorophyll, when you're detecting it, it can have cyanobacteria, but it also cannot. So chlorophyll is also another important indicator for that. And in general, chlorophyll is also a really good indicator for the actual health of the water body. Is there any other questions like that?
Like if you had a lot of chlorophyll, is that healthy or is it not healthy?
So a lot of chlorophyll, super green, is bad because it covers up the water body. So it's also limiting photosynthesis downwards the water column. It's also due to like stratification, right? So the lower parts of the water column is remaining cold while the top is warm, inducing these chlorophyll blooms. It also kills a lot of fish or any aquatic plants that are within, because it also, due to the limiting photosynthesis, also changes the oxygen levels in the water column. So yeah. So from here, just to transition. So my first paper is titled Remote Sensing of the Spatial Variability and Concentration of Chlorophyll-a in a California Drought Impacted Lake. Very lengthy. And this is looking specifically at San Luis Reservoir. So San Luis Reservoir is the 5th biggest reservoir in California. And it's also located in the Central Valley. So the overall questions for this paper is, what are the bloom dynamics in a drought impacted reservoir? So the images I've included were actual field campaigns that I led during last year and also this last May. So you could see through like the gray delineated lines outside of the lake that the water levels are very low, right? But in this last May, you could see that the water levels went back to what we would say like normal size. And the white crosshairs are where I collected my grab samples. I forgot to add them in the last image, but as you can see, it's going with the same pattern as all the other images. So the objectives for this paper would be to create contingency tables based off of World Health Organization's their standards for chlorophyll-a, and determine how lake volume influences the spatial variability of these blooms, and to identify which chlorophyll algorithms actually perform better in different chlorophyll-a concentrations in California. So my first hypothesis is that in a drought impacted reservoir, warmer temperatures, as well as I previously mentioned, the thermal stratification will lead to increased blooms, As well, when lake storage is low, this will create the nutrients being more concentrated at a lower lake volume. So this is in the Central Valley. So the surrounding soil is primarily used for agriculture. So most of the runoff is agricultural runoff, which as also it's known to contain a lot of nutrients. So this is why this lake is infamously known for its algal blooms. So the overall approach for this, I'm starting in 2017 because as shown in the previous table for Sentinel-2, it was launched in 2016, but in terms of for the water year, we're starting with water year 2017. So all of these images are taken in August. I want to show that the lake is not always super low, as shown in my previous field campaign. So these are all taken in August. We could see that there's differing lake levels across the years. It's just that this last year and also in 2018 and 2019, you do see that the lakes has lower storage levels. And the overall approach, again, as mentioned, looked from 2017 to 2021 and use different Chlorophyll-a algorithms in order to see which to detect. And I'm totally sorry, I have Landsat 8 here. So please pretend like I crossed that out. This is just for Sentinel-2. And we will also just general analyze the frequency and concentration and the spatial variability of chlorophyll in these blooms. And again, determine if certain areas experience more frequent and stronger blooms according to the World Health Organization standards. And in the bottom right was actually my first day of the field campaign for the collection of chlorophyll. And you could see it literally looks like a thick mat of grass. So I will briefly mention later on that this lake is notorious again for algal blooms, and not just that, it's really hard to accurately measure the algal blooms, not just in the lab, but also through remote sensing, just to how thick these chlorophyll grab samples are. So here I have a graph showing San Luis Reservoir from 2013 to 2022 or 2021 water year. In black, I'm showing the lake storage level. So you see that it's been oscillating throughout this time series. And in the colors are the California Recreational Health Advisory risk for cyanotoxins. And this data is derived from the Department of Water Resources. So we can't say with complete confidence that lower lake levels lead to high advisory. We could see both situations with higher lake storage and lower lake storage, and the same can be said with no advisories. But the reason why I even provide this image is to show how much data gaps there are. So in the beginning years, we only see that they're only collecting 2 to 3 times of that year. And then with greater funding, and of course, as I previously mentioned, with more public opinion regarding these issues, we're able to see more frequent monitoring. But even with then, it's only monitoring during peak season, which typically begins around May, whenever those that work in San Luis Reservoir, when they report, they start to see a bloom. And it runs until like October. So nonetheless, because of these data gaps, remote sensing, again, is a monitoring tool. It's not suggested to replace lab collected data, but it helps address these data gaps because, again, even if you're just monitoring just straight chlorophyll or cyanobacteria, it will at least help with these advisory levels since we're not sure what's going on or the patterns in between these data gaps. So again, I keep on mentioning these WHO standards. So here I am just outlining what they actually are. So for a low advisory level, it's greater than 0 micrograms per liter up to 10 micrograms per liter for chlorophyll. And there's another measurements for cyanobacterial cells over milliliters. I have not collected that because my wet lab is not equipped to measure cyanobacterial cells. So for this paper, we're only looking at chlorophyll. And for moderate, it's 10 to 50 micrograms per liter. And for high, it's over 50 micrograms per liter. So this is derived from the WHO standards from 1999. There could be a lot of conversation about whether this is pretty outdated. if the limited bins really is a good indicator of overall blooms. But again, this is probably the most common and popular method of showing chlorophyll in terms of public health risk in current papers still, even though it was released in 1999. Whether it should be updated or not is not to my discretion, but I just wanted to put it out there in terms of my mapping for my future graphs. So here is like my starting point. So I'm using an atmospheric correction method termed acolyte. And this is removing the atmospheric effects shown over the lake, which is very important over especially water bodies due to the scattering of the water like and gas like particles like across the land water interface. And I'm masking this using the modified NDWI. And in the last output, I'm using that acolyte image and using it for the 3 chlorophyll algorithms that I will talk about in a second. But you could see how these swirls and like from these images, how it outproduces to scale bar for these chlorophyll levels. So here are some actual results. So these pictures are from my field campaign in the same order as previously. So the colors are again determined by the WHO index. So if you see white within the lake, that means that there's no advisory. So that means it's 0 chlorophyll. Green is if it's up to 10 micrograms per liter. If it's yellow, it's 10 to 50. If it's red, it's over 50. And on the left here is what we have our contingency table, or also known as a confusion matrix. So across in that blue it gives you the overall accuracy. And for this, it's 51.4%. And above that, where you can see on this specific image slide where it's 3, 5, and 1, that is the false positive. So 25.7. So in this case, the false positive means that the remote sensing products are actually overestimating chlorophyll. And with the false negative being 22.9%, which means that it's actually underestimating chlorophyll. And again, it depends who you talk to. What would you actually prefer if the remote sensing products overestimates chlorophyll or if it underestimates? But of course, you'd want the overall accuracy to be better. And this is kind of where I was going off of my off tangent. These bins are pretty limited. Some of my lab in situ like data were off by less than a microgram per liter, but it would be set to the next bin. So even though it's only overall accurate from 50%, using the WHO standard protocol, not sure if that's the best way, but in current practices, this is the best way of doing it. But from remote sensing, this is like another question to ask of whether that's appropriate or not. So second chlorophyll algorithm is Mishra. So we see a similar pattern in that the overall accuracy is still around 50%. However, we do see that it is the false positive and negative are slightly different in that this had higher false negative, I believe, than the previous image. And ultimately for Moses, we actually have greater false positive. So it's more overestimating the chlorophyll levels and actually has a less overall accuracy. Again, it depends on what you value more. If you'd rather your system to be overestimating chlorophyll rather than underestimating, and that your opinion would see which chlorophyll algorithm works best. So in terms of answering the question in terms of spatial variability, I've created some semi-variograms. So this is using the first chlorophyll algorithm GONS. So this is like throughout 1 year. And you could see that there's a lot of spatial variability. So in terms of these semi-variance graphs, we're not able to really see that there's a certain area or there's a certain part of the lake that's consistently having the same amount of chlorophyll concentrations, right? Especially when we could see it on the semi-variograms on the right. We do see in some that like there may be able to like predict some spatial or some chlorophyll blooms in certain areas, but throughout the year, it's not really predictable. And it could be even more said with Mishra algorithm and as well as Moses. It's much more random and it's not really predictable in terms of looking where you can see these algal blooms. So the overall outcome is, again, to see how the spatial variability of chlorophyll is within this very, very impacted lake. And again, to see which chlorophyll algorithm best performs in this type of lake, because again, these algorithms are built in other systems. Most are created in Europe, so they're in different climate zones. And of course, also to support the validity of using remote sensing for this type of work for future papers. And also ultimately to create cautionary advisory areas if found that there are certain parts of the lakes that experience more frequent and more concentrated blooms. So paper 2 is now again talking about wildfires. So the previous paper was more for setting up the stage of using remote sensing for water quality. While here, now we're looking back into wildfire, and now we're talking about specifically cyanobacteria. So the 3 overall research questions that I'm asking is, what's the influence of wildfire regimes on lake and reservoir water quality? And do these lakes share similar watershed characteristics? And what's the lag of these water quality responses? And again, if this lag is also determined by similar watershed characteristics. So the objectives is to look at water quality trends with lakes in watersheds that have experienced wildfire and evaluate the influence of different wildfire regime variables on water quality and determine watersheds with similar climatic or environmental characteristics and to see how that determines the actual influence by wildfire. So as previously mentioned in the beginning, that the primary reason why we would expect these water quality changes is actually through the introduced sediment that's predicted. So there have been papers that have modeled to see how sediment will move throughout the West. And as you can see that there are greater mobilization of sediments through erosion, and that's typically due to increased precipitation, right? So the mobilization of these sediments in nearby water systems is what's predicted to create these water quality changes, whether it be through direct turbidity, which again is the clarity of water, or through eventual chlorophyll or cyanoblooms that again is through the introduced nutrients that's also transported through these sediments. And again, the key factors are the timing and the magnitude of these precipitation events after a wildfire. So my overall hypothesis is that more frequent fires, and if there's a higher proportion of area burned with a higher fire severity in that respective watershed, will lead to increased cyano harmful algal blooms, or also dictated as cyanoHABs. And it would also lead to a higher cyanobacteria index value. And this is due to the increased soil erosion potential, which would lead to charred vegetation and surrounding soil to increase the nutrients in the water body system. And again, as previously mentioned, through the incoming sediment, this is because there's often a water repellent layer that's formed above the soil surface in high to moderate wildfires. And so this hydrophobic layer actually leads a lot more than expected runoff on the surrounding area. So with an increased amount of runoff, we would also expect an increased amount of sediment entering nearby water inlet systems. So overall approach, again, as previously also supported, in situ data is very important in all of remote sensing. So I've created an in situ database for all of California. So these are my delineated watersheds, and I'm using it in a HUC 8 approach, simply because in a if a hug 10 is a bit overestimating where I'm capturing too many lakes while going to, sorry, the opposite. If I'm going to a lower hug lake value, I'm overestimating. While if I'm going to a higher value where the watersheds are smaller, I'm actually dividing up a lot of these wildfires and it won't be really indicative. So my perfect medium is in a hug 8 level. I've collected available water quality data for the whole state for Chlorophyll-a, Cyanobacteria, and turbidity dating from 1984. It is very limited. I will admit that this map seems that like I've collected a lot of data, but again, it's very limited due to either budget restraints or actual like feasibility of actually entering these water systems. But I've already done all the work in collecting what is out there. And again, this is where the big data factor comes into my actual presentation. So for cyanobacteria, you can only look at years from 2002 to 2012 with the launch of NBSAT's Merced. And then there's a data gap because of Merced's, like it got shut down. And then the launch of Sentinel-3's Ocean Land Color Imager was launched in 2016 until now. So these data go by like a 10-day rolling max, and they're actually able to pick up data every other day. So it's a lot of data for about almost 2 decades worth of data. So I have images from each of those dates for the whole year. And I'm also summarizing through MTBS, the fire frequency, the extent, and also the difference normalized burn ratio. And also other wildfire regime characteristics such as like the return interval, as well as other watershed characteristics such as the vegetation type, the post-fire erosion potential, which is also provided by CAL FIRE, which is California's like local wildfire system, and the MRLC land cover data, which also covers tree canopy and also the impervious surfaces of the watershed, and also MERA2 climate data, which is basically a modeled climate data by NASA. So summarizing it all through a HUG 8 watershed level, and then relating it back to that specific lake and seeing if there are any water quality changes after a wildfire. So this is an example of how this data actually looks. So this is showing a cyanobacteria index for Clear Lake, which is also another heavily impacted algal bloom lake. And you can see how we get the overall cyanobacteria index level spread across the lake. And we're creating a time series basically every other day. So this is thousands and thousands of files. And so, and where to limit the actual relationship of what would be like through post-fire. From what I've read online and then through like all the periodical or peer-reviewed journals, it's typically up to 5 years. However, 5 years, I have a little asterisk there because you're letting a lot of error to come into account if you lead to a longer time series due to other environmental or just climatic effects to interfere with the wildfire response. So I'm limiting it to 2 years, which is also commonly supported in the literature. And again, also identify which watersheds appear to have a similar lag response time in terms of the cyanobacteria results across California. So the outcomes of this is that it will show watersheds that have greatest amount of water quality change post-fire and identify a range of lag responses in different watershed properties. And at the end of my talk, I actually have like a couple of columns, or not a couple of columns, a couple of rows actually of like what my current data set looks like, just so you can see the amount of columns and data that I'm going to be summarizing in order to address this question. So paper 3 is talking about now turbidity and chlorophyll, which was previously mentioned. So the research questions are identical to paper 2. They're asking the exact same questions. However, they're looking at different water quality variables, and now it's actually with satellites at a finer resolution. So again, this uses Landsat and also Sentinel-2, which uses a 30-meter and a 20-meter resolution respectively. And again, the objectives are very similar. However, this is again through multidecadal. So Landsat was launched in 1984, which also why earlier in the fire graphs, it started from 1984. While the Cyanobacteria only starts from 2002 and with that big data gap in between. So we're actually able to look at the time period much longer to see if these wildfire characteristics are changing over time as well, along with the changing landscape that's summarized with the Hug-8 watersheds. And again, similar hypothesis and rationale that in areas that have more frequent fire, greater fire burned, and a higher severity, we'll see a higher change of water quality in the respective lakes. And for the approach, Landsat would only be used for turbidity because it lacks a spectral band for accurately capturing chlorophyll-A, even though there are some papers that do use chlorophyll-A for Landsat. Sentinel-2 would be only used for chlorophyll. It does have a much more limited time restraint for this since it's been launched only in 2016. So you can only be looked at for, for a couple of years, while turbidity can be looked at a longer, much more longer time period. And here's kind of like showing what I've been talking about. So in the dark red on the top is Landsat 5, but we could still use turbidity. And Landsat 7, its launch was while Landsat 5 was still out in use. And so NASA, it has a revisit interval of, instead of every 16 days, it's actually every 8 days, because these two satellites were programmed in a way that it's able to be used both in synchronous over an area. So it's not like it's going over at the same time, rather it's actually cuts the revisit time in half because of it. And then of course Landsat 8 was launched and Landsat 5 decommissioned. So here you see the time series for turbidity. And for chlorophyll, it's only limited to, again, from 2016, as shown in the next. And this is just mostly talking about how, even though Landsat has shown some papers that it could detect chlorophyll, and in terms of my paper, I'm not going to use that since it lacks the red edge band that Sentinel-2 has. That is better to detect chlorophyll-A, since the red edge band is able to detect chlorophyll-A rather than confuse it with color dissolved organic matter, which is CDOM, which Landsat typically does. And the overall outcomes, of course, is to observe the variability of water quality over the years in a changing climate. And we're going to be able to see the relationship, if there is one, with wildfires and water qualities, and to see how these watershed properties influence these changes. And that's the end of my presentation. I do want to give, give me just a second. This is updating right here. Here you go. Oh yeah. I do want to give a special shout out to NASA. This is done in a project that's been funded for until 2024, so for 3 years. I'm a Finesse Award recipient, so this overall big wide data system has been shown to, has been deemed important simply because this question hasn't been asked before, and especially at such a large scale. So I know it seems a bit murky in between, but when you look at my actual data table, you'll see why. I'm still kind of all over the place. And here are my references. So let me pull up the data frame that I'm talking about.
And Brittany, you're being
Yes.
very humble. this, like, people have studied fire and watersheds. They typically study like one watershed at a time, and it's very hard to make inferences. So Brittany's approach to look across all the watersheds in California over decades has never been done before. So that don't be so humble.
Like, that's why it's like, I did my chlorophyll work at San Luis after I started my cyano work, and simply because I had a funding opportunity for that. So I haven't touched my cyano work until I literally came here to work with Natasha. And so much data has been updated where I've had to re-download everything and basically run all my program from scratch because it ended from 2019. And so since the end of the pandemic, a lot was not worked on, and now everything was published. So now I have to update everything. So here in blue, I've color coordinated for watershed characteristics and also
I think you're gonna have to stop sharing.
and re-update here.
Yeah,
Yes,
yep.
you're right. You are absolutely right. I'm gonna stop share and do it for this screen. Go. Yeah. There. Oh, no, it's still not.
It's
Okay.
sharing on here.
Is it?
Yeah.
Oh, okay.
On Zoom it worked.
All right, okay. Let me check that then. I'm just gonna exit out of PowerPoint. That's probably what it is.
So a large part of why Brittany is here is to figure out how to build this database to do this analysis. And it's still very much in flux. So like, I think Brittany, one of our ideas for today was to have a discussion, an open discussion. So we're like really hoping that you guys all have ideas for us, so don't be shy
Right. Yeah.
about sharing your thoughts.
Here you go.
Yay.
Finally. Okay. So in blue are watershed and lake identifiers. So the TNM ID is the identifier for a specific watershed, and that's like San Francisco Bay per se is the name, the common name for it. We have area acres for it, the size of the watershed, and here's the lake name. So Calaveras Reservoir is within San Francisco Bay watershed in the lake area. And of course, here we have the date. So I do want to emphasize that, this is just the first 10 columns of my data set. And for cyanobacteria, again, I have basically every other day. So I want to actually show differing numbers. So this is summarized by month rather than looking at it by day, just so we could see differing numbers along these columns. And in this type of cyan looking color are the actual cyanobacteria summary variables. So I'm looking at it at both, as you can see here, a full pixel and SCI approach. So when I say by full pixel, is that I'm looking at the raster data set as a whole. So I'm looking at pixels that are not also cyanobacteria. So in this case, I'm saying like, okay, the median full pixel is 0. So even accounting in areas of the lake that has shown no cyanobacteria, the median result is 0. However, that doesn't mean that there's no cyanobacteria in that lake. But as we can see, like in this row, right? We could see that if we're filtering out the parts of the lake that has no cyanobacteria, the actual like median, or actually, sorry, median is still 0. But like the maximum is 133, right? Like that's like how I've been summarizing it, both looking at it as a whole. So seeing the pixels that don't have cyano and also just looking at purely the cyano pixels. And here in orange are the fire regime variables. So total fires within that watershed for, in this case, in that given month. And the fire extent.
Can you possibly zoom in?
Yeah, of course.
To make it a little bigger.
Let's see if this helps. Because this is
There's just like up in the top left, there's the zoom from 100, you can move it.
Oh, right, okay, great, yeah, just because it ain't mixed up.
Much better, thank you.
Oh, yeah, this is fine. Okay, so yeah, in orange, the wildfire regime variables, so again, a fire return interval. and also looking at the MTBS DMBR results, looking at the mean and standard deviation of this, and also looking again at the land class. So the national cover of the national land cover data set. This is updated every couple of years. So when I'm doing these results, I'm matching it to the closest year for that. So as we could see that it's changing given the differing years. Same thing goes with the impervious land surface. So impervious land surface means that like if it's more urban area, so more urban area in general, impervious surface, that means the water can't percolate within the water column. So that might lead to extra runoff, and compared to an area that could absorb that precipitation. And again, similar variables to like the cyano index and tree canopy cover, saying how much vegetation is in the area. So the MRLC only has 2 data sets for this. It's only in 2011 and 2016, so it's much more limited. And WHR is actually looking at like the vegetation type, like in that area. So the majority of that is urban, as you could expect in that certain type of watershed area. And in gray, we have a summary of climate results. So for that month in that watershed. So precipitation, the wind speed, and also the overall air temperature. And in red here, oh, sorry, they should have erased that because it's already mentioned. So in red here are actually the 3 variables that I'm still really trying to work into and still ask myself. So fire rotation, if that's really indicative or actually useful in my watershed analysis, and the cumulative distance of fire from the watershed, and also the shorter distance of the watershed. I'm still working into that because of what you could imagine of how these river and stream networks work. How would I indicate, like, how do I actually summarize and determine how far these fires are from a lake, right? Like through what streams? Because even though there's the shortest path, it doesn't necessarily mean through runoff that that's how it's going to enter. Like there could be differing slope into context, or if there's actually something in the way. So these networks of like flows is something that I'm still working into for my overall data set. And yeah, that's basically it for what I have.
And Brittany, just to
Yes.
add, because I think you missed a point of clarity here. So what we've been doing with this data set is that like each of these rows is like a cyano event. And then we're saying, okay, from that cyano event, like what was the burn severity in the last 12 months? What was it in the 24 months previous to that? So that's the
Right, yeah, I'm so sorry, yeah, because just for like the context of this, so I could have like differing numbers. So I previously mentioned that this is summarized by month. So for the orange and where I have the mean NDVR for MTBS and the standard deviation. So I wouldn't be looking at it per se like for that month. I'd be looking at it from the past 12 months, from the past 24 years. So like previously mentioned in my presentation, I'm looking up to 2 years, but I'm also seeing like what's going on earlier than that, just for 1 year immediately after as well.
That's for any fires within that particular watershed.
Exactly,
Gotcha.
yes. And there are some fires that overlap multiple watersheds. So like some of that would be repeated.
Or would you break up the fire by watershed? So you'd say this much of the fire happened in this watershed, this much of the fire happened in this other watershed.
Right, exactly. So like actual, like another, I don't think I have it here, nor have I included it before, but I think it was once mentioned, the actual proportion of the watershed that was burned in terms of all of this as well. Again, this is like, I think it's like 50 columns. So like, I think like every other month I add something onto it. At some point, it's just like, where do I stop? But yeah, so I think I see something in the chat. Oh, my total bad.
Yeah, I have a thought on the ones you're still thinking about in the red. Doing some sort of like, well, at the simplest form, like you put in distance to burn pixels. you know, or even the fire edge to look at, you know, and then you overlay your stream network and you can get sort of a value of like how far away burned areas were. But I also think like combining that with some sort of like topographic measure, like there's the height above the nearest drainage or hand. And so that just like counts like how far away from a drainage you are. So I can see like some combination of like Euclidean distance and a height above the nearest drainage to be pretty useful with the last two there.
Yeah, thank you. Yeah. I've also considered like using like, okay, why don't I just use like a DEM? But it's like this DEM are producing these river flows. It's like I can't just do that. But yes, thank you so much for like
Yeah,
mentioning
Ty
that.
gave some suggestions.
Yeah.
And Matt Rossi gave some suggestions of some packages that exist. So.
Yeah.
And then you would do all the math internally to the network. Since they're internal to the network.
Speaking of Matt, he has his hand up.
Hi, really nice talk. Thanks for sharing that with us. Yeah, I was just thinking about that question a little bit. I mean, one of the things that you might consider is that even in your kind of California transect that you're looking at, your lags may vary a lot from Southern California to the Sierra and Northern Sierra. So like in the Pacific Northwest, we actually think the peak erodibility of the hill slopes is maybe 4 to 7 years after a fire, as opposed to 1 to 2 years after a fire in maybe Southern California. And so you might expand that range of lags you're considering. It's because when you have roots in the soils, they actually stabilize the soils even after the trees have died. And so there's a time of decay that actually dictates kind of the peak loading of the systems. But yeah, there's lots of tools you can use, and I know Ty gave you other tools as well to start thinking about those distances, travel distances. But one thing I would encourage you to think about is actually like, where are channel heads? And so the distance you might care about is what's the distribution of distances from your fire kind of domain into the closest channel head. Because once they get into the channels, they move very fast, and getting into channels, it usually takes longer depending upon the process. So those are my 2 thoughts on that.
Thank you. Actually, just to like briefly go off of the longer time period. So for cyanobacteria, I have constrained that for 2 years, just given the data gap and also the limited range. So for my talk that like goes back to 1984, I for sure could do that for looking at it a longer time series. I think like I've just narrowed it down to 2 years, just simply like I'm not able to look at longer time periods for the limited temporal data availability I have for this. But for the other ones, yes, for sure. That's also another reason why I'm using remote sensing, right? Like the data is actually there compared to actual field campaigns that can only look at it for 1 to 2 years post-fire. So
are
yeah.
you planning to make this table for both the turbidity?
Yes.
And like each of those water quality metrics?
Exactly. It would literally like whatever I'm basing it off of this, I would just make it into back to those years and changing the cyanobacteria and like outcomes and switching it for turbidity. It's exactly how it would be.
I was also going to add that I really like that you're trying to include the watershed properties source areas, but one data set you might look at in the reservoirs themselves is each of those reservoirs has a rating curve, which says what the relationship is between volume to water depth. I mean, volume to water depth. Yeah, that's right. And so it might be that your most vulnerable reservoirs are actually those where you have a lot of shallow water depths, and they become more vulnerable as they dry out. And so it might be a property that you can extract directly from the gauges that you get there, where they have rating curves. They know what that relationship looks like. And it may be a property that's even more important than the source area in terms of how vulnerable you are to like algal blooms.
Yeah, no, that makes a lot more sense, and also relates back to when I was mentioning of San Luis Reservoir, the lake storage. So yeah, no, that's a really great point in including that. Thank you.
Thank you so much, Brittany. This was. I just want to make a call